Hello, in this video we are going to talk about Chapter 7, Cellular Respiration, and the steps that are included in cellular respiration and where it occurs and how it can be different for different situations. So what you're looking at here is a meme that I created with the major steps involved in cellular respiration. Now, this is the typical manner that we learn cellular respiration in. Of course, this is not how cellular respiration can happen all the time, but I will say as long as oxygen is present, these are the steps that we will take. And as long as we have mitochondria, these are the steps that we'll take. So we start with glycolysis, then we move on to the creation of acetyl-CoA, then the Kreb cycle, and then oxidative phosphorylation. You might also know oxidative phosphorylation as the electron transport chain. Another term for the Kreb cycle is the citric acid cycle. So each of these steps creates certain products, which are seen here. So first thing I want to point out is that glycolysis only occurs in the cytosol. So as long as a cell has cytosol, it's going to do glycolysis. So if we think back to the chapter where we learned about cells, what's one of the four things that all cells have? It's cytosol, right? So all cells can do glycolysis. It doesn't matter if they're a prokaryote or a eukaryote. So after glycolysis, these are the products that will be available. And then if oxygen is present and the organism has a mitochondria, then they will move on to these additional 3 steps, and these are the products that they will make at each step. So notice how different the products look from step to step. I do want to go over a few of the terms here before we move on to until we move on to each of the steps where we break it down. So first we start out with glucose. So glucose is needed to begin cellular respiration. Glucose is broken into 2-pyruvate, and then we also get 2 ATP and 2 NADH from breaking glucose into 2-pyruvate. And then the pyruvate are changed into acetyl-CoA, and this is where we can release some CO2 as a waste product. And then if you notice this third step, the Kreb cycle, it has a lot of these NADH and FADH too. So these are electron carriers. That is their major job. They capture energy and they store them in chemical bonds. And then these molecules here and here are taken to this last step to create all of this ATP. So you do see oxygen present here as a product. Oxygen acts to catch electrons. But the major goal is what? To create ATP. So ATP is our cell energy. It's very stable cell energy, and it's better than NADH or FADH too. ATP is more stable. Okay, so what we plan to learn in just 7.1, we're just going to talk about energy. So we'll talk a little bit more about ATP and NADH and FADH too. We will talk about how electrons matter in cellular respiration, and then why ATP is our energy source. Okay, so the mitochondria is very important in cellular respiration. Remember, in order to do the last 3 steps on the meme that I had, the creation of acetyl-CoA, the Kreb cycle, and oxidative phosphorylation, we have to have a mitochondria present, and we need oxygen too. So those 3 steps occur in the mitochondria. Now I do want to point out that all eukaryotic organisms have at least 1 mitochondria. Now you do have a few cells in your body that don't have mitochondria, and they can only do glycolysis, and those are red blood cells. So RBCs, red blood cells, their major job is to transport oxygen around the body after capturing it in the lungs. So they don't have a mitochondria, and they actually don't even have a nucleus. They get rid of all the extra organelles, and they only have room for their hemoglobin protein, which takes up oxygen. So that's a special case where a eukaryotic cell doesn't have mitochondria, but all eukaryotic organisms have a mitochondria. So yes, plants have mitochondria. They need it in order to create ATP. Without it, they wouldn't be able to survive. So taking a look at this, here's our cellular respiration chemical formula. So we start with glucose. So remember, glucose is a carbohydrate, right? C6H12O6. It follows that 1 to 2 to 1 ratio of carbon, hydrogen, oxygen. So glucose is not normally found on its own, right? It's usually a really long chain, which is a polymer. So polysaccharide is a very large carbohydrate. That's the polymer of a carbohydrate. So usually your body has to break the carbohydrate down into individual glucose components for it to go into cellular respiration. Okay, so this is after the digestion point, so we have just single glucoses now. Next, you'll need oxygen, so you need to breathe, which is hopefully what you're doing right now. And then our products are CO2, which you exhale, and then water, and then ATP energy. So CO2 and H2O are typically referred to as waste products, but ATP is our major goal of our respiration formula. So I do want to point out that even though this formula is written in such a simple form, there's a lot more going on here. And this doesn't happen in just one foul swoop. There are a lot of steps, as we've seen so far, that extract the energy from glucose. So if we were to break glucose down in one reaction, we wouldn't be able to get enough, or we wouldn't be able to get all of the energy from glucose that we could if we broke it down in small incremental steps. So with those small incremental steps, we're able to get the most ATP energy from glucose. Now we use ATP and not just glucose as our energy source in a cell because ATP is more stable, it's smaller, we can transport it around the cell a little bit better, and it's not huge, it's not this huge molecule, so we want to make ATP. Okay, so this is cellular respiration just simplified in the location, and I will say that the Kreb cycle is in the wrong location. It should be inside of here, but anyway, this is good enough. So glycolysis starts with glucose, creates 2 ATP, it also creates pyruvate, and then the next step, if we have oxygen and a mitochondria, enters the mitochondria here, and then acetyl-CoA is formed, the Kreb cycle happens, and then boom, the electron transport chain, which is probably the most important part of cellular respiration since we're making all of our ATP here. So let's look at it a little bit more in depth, so hopefully this doesn't scare you. Again, we are looking at a eukaryotic cell that has mitochondria in it, so this lighter portion is, again, just the cytosol. We're in just the cytosol, the cytoplasm of the whole cell. And that's where glycolysis can happen. And then in the mitochondria are the next 3 steps. Pyruvate turns to acetyl-CoA, the citric acid cycle or the Kreb cycle, and then the electron transport chain. So I do want to point out that, again, here's all of our products that we're creating. I'm pointing to all of them. But again, the major product that we're interested in is an ATP. Now, I do want to point out that the Kreb cycle or the citric acid cycle creates all of these electron carriers. And all of these electron carriers from the Kreb cycle and then those from glycolysis go into the last part of cellular respiration, which is the electron transport chain. So keep that in mind as we talk about each of these steps individually, okay? So the major takeaways, and I know this is probably weird to have takeaways at the beginning of the lecture, but major takeaways of cellular respiration is that cells extract energy from food to create ATP, which is cellular respiration. So having just food molecules, so carbohydrate molecules, floating around in cells, those aren't easy for cells to extract energy from at a ready-to-go type of basis. So we have to change them into ATP. So it's like you went from France and you brought your money with you and you came over to America. Well, now you need to exchange it so that now you have American dollars and that you can use American dollars anywhere inside of America. Okay, so that's all we're doing. We're just kind of exchanging, okay? As we work through the metabolic pathways involved, notice the close link between the transfer of energy and the movement of electrons. So if you know anything about chemistry, electrons provide our energy. So even electricity is from electrons, the movement of electrons, okay? So energy in a cell is also due to the movement of electrons. Now, again, we only have a small incremental transfer from 4 different steps, 4 major steps in cellular respiration so that we can collect energy in small packages. If we did it all at one time, number one, we wouldn't get all the energy that we could get. We would release excess energy. And number two, that excess energy would come out as heat, and that would make your cells too hot, and it would probably cook them and they would die. So it's better to have all of these different steps to take all of the energy into chemical form energy and not heat energy. So a lot of these steps are termed a redox reaction, where we're transferring electrons from one point to another. And in that transfer of electrons, one molecule is going to be reduced and the other one is going to be oxidized. So the one molecule that's reduced is going to be capturing electrons. The one molecule that's oxidized is the one that donated the electrons. Okay, so when I talk about like NADH, it has that H on the end, that's going to be the reduced form. So it's actually, even though I know the word is not that descriptive, but NADH, because it's reduced, actually has more energy, which sounds backwards, right? The reduced, you'd think it didn't, but the reduced form actually has more energy than the oxidized form. Okay, so more about redox reactions. So here's the difference between an oxidized NAD and a reduced NADH. It's that hydrogen molecule right there. So by adding that hydrogen molecule, we've created another bond, and then that bond can be broken at a later time, and energy from that bond is donated to a reaction, which is going to be in the electron transport chain. So ATP is our goal of cellular respiration, right? It's actually a type of nucleic acid. So if we think back to our 4 biological macromolecules, DNA, RNA are nucleic acids, but also ATP. ATP is a nucleic acid, so it has a ribose sugar. It has a nitrogen base, which is adenine A, and then it has these phosphate groups. Now it has 3 phosphate groups in ATP, two phosphate groups in ADP. So ADP is the oxidized form, so it's lower energy. ATP is the reduced form, so that has more energy. So we can create ATP from an ADP and a loose phosphate group. So all we're doing is putting this last phosphate group on. We can create it in two ways. One is substrate level phosphorylation, and the other is chemiosmosis. So we're going to see substrate level phosphorylation in glycolysis and in the Kreb cycle. And then we will see chemiosmosis in the electron transport chain. Okay, so most of our ATP, again, is created by chemiosmosis because that's where the electron transport chain is, or that's what the electron transport chain does. So hydrolysis is how we get energy from ATP. So cells will break that last phosphate group off. See, so this is what's happening. So we're breaking that last phosphate group off, and then in that chemical bond is where the energy is stored. So that's the energy that cells will use in order to drive different processes. Losing that phosphate group is dephosphorylation. Putting that phosphate group back on is phosphorylation. And phosphorylated molecules actually tend to be less stable and more likely to react, which is kind of a good thing. So that means it's easy for cells to break that third phosphate group off and get the energy from it. Okay, so the energy currency used by cells is which of the following? Hopefully you said ATP. Oops, sorry. Hopefully you said ATP, which is A. So that's our high energy form. Reducing a chemical reaction is called what? Or is what? It's by adding an electron to the substrate. So remember, reducing is the higher energy form. So if we have more electrons, we're going to have higher energy. We're also being reduced. So I think like electrons are negatively charged. So if you're adding electrons to a chemical, to a molecule, you're causing that electric charge to be reduced, which is the reduced molecule. Okay, so that's 7.1. So hopefully now you understand what ATP is and how we can change ATP to get energy from it. So we can phosphorylate it. We can dephosphorylate it to get ADP, and then we can phosphorylate ADP to ATP to get to the higher energy form. But of course we need energy to do that. Hopefully you know why NADH and FADH2 are important. They're electron carriers, and hopefully you understand what reducing and oxidizing means. Okay, so we're going to talk about each of the steps of cellular respiration. We're first going to start with the first step, glycolysis. So we will talk about the overall process and different the results of glycolysis. I do want to point out that these are the reactants of glycolysis, and these are the products of glycolysis here. So if you notice, we need 2 ATP to start, and we get 4 ATP to finish. So that's a weird thing about glycolysis. But anyway, so let's take a look at this again. So here's our formula, again, written out. Glycolysis, again, is the first step. Glycolysis occurs in what? The cytosol of the cell, right? So remember, all cells, no matter prokaryote, eukaryote, have cytosol. That's one of the 4 major characteristics that cells have to have in order to be a cell. So all cells have cytosol, so all cells can do glycolysis, okay? So glycolysis, again, is the first pathway of glucose metabolism. So it does have a lot of reactions. It has 10 enzymatic reactions. So there's 10 different enzymes that act on glucose to change it into pyruvate, and we get some other things out as well. So oxygen is not required for glycolysis. Glycolysis is referred to as an anaerobic process because it can happen in the absence of oxygen. The inputs, again, you put in one glucose molecule here, which is a monomer of carbohydrates. So your body has already broken down major carbohydrates into small portions of glucose. We also need the oxidized form, NAD. We need ATP to start. We need to put 2 in to get some more out. And then we need the oxidized form of ADP. So ADP, NAD, these are the lower energy forms. The outputs are 2-pyruvate, which is basically glucose cut in half, is what 2-pyruvates are. We also get the reduced form, NADH, so higher energy. We get the reduced form, ATP, and we get some oxidized ADP. So we get a net gain of 2 ATP, and then we get these 2-pyruvate, which move on to the mitochondria if available, and then these NADH, which move on to the mitochondria if available. So glycolysis, again, the first half of glycolysis uses 5 enzymes, the second half uses 5. There are a lot of steps involved. We are not going to learn them, but I will show you. Okay, so 1 through 10, these are the steps of glycolysis. Again, this is what's happening in the cytosol. So all of these enzymes, hexokinase, all of these, are floating around in the cytosol of the cell, ready to do their specific reaction. So proteins at work, right? Enzymes. So you can see where we use the ATP. We use the ATP in the first half of glycolysis. And then in the second half of glycolysis, we start to get our products, which is NADH, ATP, water, and more ATP. So the net gain, again, is 2 ATP. We also create, which are also equally important, 2 NADH and 2-pyruvate. So these are going to move on to the mitochondria if available. Okay. So glycolysis realizes it produces way less ATP than oxidative phosphorylation, but at least it's honest work. At least it's something. So it's some energy. There's a nice video on YouTube if you want to learn about the different steps, the enzymatic steps of glycolysis, but we are not going to learn the steps. So during the second half of glycolysis, what's happening? What do you think? Well, letter C, right? ATP is made. So we use ATP in the first half. So if I asked you during the first half of glycolysis what happens, we would say A. But since this is the second half of glycolysis, we're saying C because ATP is made. So in summary of glycolysis, it occurs in all types of cells, prokaryote, eukaryote, plant, animal, fungi, protist, all of the above. It occurs in the cytosol, and oxygen doesn't really matter if it's present or not present. It's going to happen either way. What it requires to begin are 2 ATP, a glucose molecule, and 2 oxidized NAD molecules. And then it creates 4 ATP, 2-pyruvate, which is basically a glucose cut in half, and then 2 reduced NADH. So the pyruvate and the NADH enter either aerobic or anaerobic respiration. We will move on to aerobic respiration, so that's what occurs when we have oxygen. But first, let's sum up our objectives. So hopefully now you can describe the overall result of glycolysis. So hopefully you know the products, and hopefully you can compare ATP and NADH input and output. Okay. 7.3, we will talk about the oxidation of pyruvate in the citric acid cycle. So in other terms, we'll talk about the creation of acetyl-CoA and the Kreb cycle. Okay, so these cycles have different terms depending on who you talk to. So we're going to talk about how the citric acid cycle is a circular pathway, which is very strange in terms of living things. And we will talk about how pyruvate is going to be prepared for the citric acid cycle and why it's important. So again, glucose is our molecule that we are extracting energy from. So we're continually breaking glucose up, and every time we break glucose, we're releasing energy. And we're capturing that release in other molecules, which are typically going to be electron carriers, or ATP, but not in this step. So pyruvate, which is typically half a glucose molecule, is going to enter the mitochondria. Now, once the pyruvate enters the mitochondria, there's an enzyme that causes it to release some electrons and a CO2 molecule. So that enzyme is now attached to that pyruvate, so now it's acetyl-CoA. So now it looks like this. So we lost a C and two O's, right? We lost those, but we still have this molecule here that we can extract energy from, which is great. So those are the 3 steps of acetyl-CoA formation. We have 2 pyruvate, so this happens twice, okay? So this happens for each pyruvate molecule created from glycolysis. So we need 2 pyruvate, we need 2 NAD, and we need 2 CoA molecules, which are an enzyme. So our products are CO2, which is a waste product. So every time you exhale, you can think about the formation of acetyl-CoA. We also create 2 NADH, which go on to the final step of cellular respiration. And then we make 2 acetyl-CoA, which are going to enter the Krebs cycle. Okay, so let's take a look again at our big map. So here's our glycolysis step. We created pyruvate. Pyruvate entered the mitochondria. So remember, we're making 2 pyruvate from glycolysis. So this is happening twice. So 1 pyruvate molecule, acetyl-CoA, attaches, that releases CO2. And then now we have acetyl-CoA, which enters the Krebs cycle, or the citric acid cycle. Okay. So we're going to, now since we're entering the citric acid cycle, we're going to break acetyl-CoA up as much as we can to get as much energy as we can from it. And then after that, acetyl-CoA is pretty much gone. Okay. Okay, so the citric acid cycle occurs in the mitochondria. It happens in the matrix. It has a lot of steps, and it is a circular metabolic process, which is interesting that it's not linear like most. So we start with acetyl-CoA, and then we end with oxaloacetate, which acts on acetyl-CoA. So that's how it's circular. What you will notice is that, I want to point this out quick, acetyl-CoA has a few carbons on it, and then we release those carbons at certain points throughout the Krebs cycle, which come out as CO2. But the major goal is that we are rearranging and breaking this acetyl-CoA molecule. So look, it looks different, looks different, looks different at each step. And every time we change that acetyl-CoA molecule, we're getting some energy from it. So we're we're reducing NAD to make NADH. So that is a higher energy molecule, right? An electron carrier. Here we're making it again. And then we're making something called GTP or ATP. Most of the time it's going to be ATP. And then we are going to create FADH2, which is an electron carrier. And then we make NADH here. So remember, we made 2 acetyl-CoAs in our acetyl-CoA formation because we started with 2 pyruvate. So for 1 acetyl-CoA, it goes around once. But since we have 2, so 1 glucose molecule makes 2 acetyl-CoA, right? It goes around twice, okay? So through a series of reactions per 1 acetyl-CoA, this is what we get. But since we have 2 acetyl-CoA, this is our final product for 1 glucose. So we made a lot of electron carriers, which are great. Those will move on to the final process. We released a lot of CO2. So the carbons in glucose are actually just released in a gas form, CO2. And then we made 2 ATP, which is not a lot. So the Krebs cycle, the citric acid cycle, its goal is not to make ATP. It's nice that we get 2 from it, but it's not its major goal. The major goal of the Krebs cycle is to make these guys that we can move on to the electron transport chain. Okay. So again, here's our Krebs cycle. Here's our carriers. They're going to move to here, which we're going to get to next. So to this point, so we did glycolysis, we did acetyl-CoA formation, and then we did the Krebs cycle. So to this point, this is what we've made. We made 4 ATP, 2 from glycolysis. We have a net of 2 from glycolysis, and then 2 from the citric acid cycle, the Krebs cycle. And then we have 6 CO2, 4 come from the Krebs cycle. And then we have 10 electron carriers and 2 more electron carriers. So they just hold on. They actually have 2 areas to add hydrogens than 1, which NADH only has 1. So FADH a little bit more powerful, but we only make 2 of them. Anyway, so that was just extra. You don't need to know that. So glucose at this point is completely oxidized. It's completely ripped apart. There's no more energy that we can get from glucose. However, we can now take our electron carriers and make all of our ATP. So the major goal, again, to this point, is to make electron carriers. Okay. So is most of the energy from glucose stored in ATP, CO2, or electron carriers? Well, you should say electron carriers. Hopefully I've really drilled that into you so far. This is one review method that I like to do with my students. I call it a 1-minute paper, but I give them more than 1 minute. So the major thing to write about that I want them to summarize is to describe what cellular respiration is, what the purpose and goal are, what the first process of cellular respiration is, what happens to glucose and what's produced, and then why a Krebs cycle is important even though there's only 2 ATP that are made. So that's something that you could do on your own if you want. It would be a great summarizing tool. I know that there's a lot of information we covered up until this point, so it might be good for you to summarize before we get on to the last few steps here. So one more poll question, or a couple, I have 2 more. How many NADH molecules are produced in 1 turn of the citric acid cycle or the Krebs cycle? Well, it's 3. So we make 6 per glucose molecule, but we make 3 for 1 turn. Another question, what do the electrons added to NAD do? Well, they go on to another pathway for ATP production. So they go on to this final step. Okay, so now we've covered the formation of acetyl-CoA, oxidation of pyruvate, and the citric acid cycle, which is the Kreb cycle. So hopefully now you can tell me the reactants and the products, so what goes in, what comes out of each of these, of both of these, and then hopefully you can tell me the major goal of each of, or of both of the reactions. So the oxidation of pyruvate, the major goal is to get acetyl-CoA, so that acetyl-CoA can enter the next step. The major goal of the citric acid cycle, the Kreb cycle, is to make those electron carriers. Okay. All right, so let's talk about the electron transport chain, aka oxidative phosphorylation. So we're going to talk about how electrons move through the electron transport chain, and we'll talk about the energy levels and how an electrochemical gradient helps to create ATP. In oxidative phosphorylation, most of the ATP produced are produced here. So this is actually the only pathway where oxygen is used. So remember, oxygen is one of our items that we need to start aerobic respiration, but we don't actually use it until this final step. The pathway here consists of an electron transport chain with different proteins, and then the final portion is chemiosmosis, which makes ATP. So this is going to be different. Chemiosmosis is different than the way that we've been looking at so far for making ATP. Normally we've been breaking chemical bonds and capturing that energy to make ATP. That's called substrate level phosphorylation. But in chemiosmosis, we are going to be moving chemicals across the membrane, and that will provide enough electrochemical energy to make ATP. Interesting stuff, right? Really physical, chemical type of stuff. So this gradient that we make with the electron transports, that's going to be enough, and that's going to power our chemiosmosis. So here's our location that we are at now. Here's our electron transport chain and our chemiosmosis. So notice that it is on the folds of the mitochondria. So if you look at a mitochondria, it has a lot of folds in there, and that increases the surface area. So an increased surface area with more membrane space, we can do more electron transport chain processes, okay? So on this membrane, we have all of these proteins embedded and these proteins embedded, and they just repeat after another all the way through, okay? The electron transports are going to pump hydrogen ions into the intermembrane space, and that creates a positive charge in the intermembrane space. In the center, in the matrix of the mitochondria, it's relatively neutral. So now that the matrix has a neutral charge and the intermembrane space is positively charged, well, we need to allow those hydrogens to come back through. And so they do actually come back through, and they come back through these ATP synthase proteins. And as they come through, they create ATP. Now, you might say, well, why do they need a protein to come through? They're so small. Wouldn't they just be able to squeeze through the membrane? No, they can't squeeze through the membrane. Remember, membranes cannot allow charged molecules through. So they're locked up here in this intermembrane space. There's no way for them to exit until they reach a ATP synthase molecule. or ATP synthase protein. So as that hydrogen goes through ATP synthase, that provides enough electrical energy to put a phosphate onto ADP. So now we have ATP. Okay, so the way that we get hydrogen molecules into the space is through these transporters. So let's take a look at these transporters. There's a few different ones. There's 5, technically 6, and they break our electron transport or electron carrier molecules, and they provide energy when they are broken, which provides enough energy to pump a hydrogen through. Okay, so let's explain that again. So our electron carriers, which we got from glycolysis, and we got electron carriers from the Krebs cycle, those are now coming to these protein molecules on the membrane of the mitochondria. So as we break the electron carriers, we break the chemical bond that releases energy, and that's going to push a hydrogen molecule against the concentration gradient into this intermembrane space. So this is where we're going to have a high concentration of hydrogen molecules. So you might say, hey, that sounds really familiar. Well, yeah, you've learned about it in the previous chapter. It's called active transport. We're just doing it a little bit different. We're not using ATP. In this case, we're using NADH and FADH2. So we're using active transport in order to pump hydrogen ions against their gradient, and we're putting them in a high concentration space. So notice all of the broken molecules here. As one is broken, it pumps a hydrogen molecule through, and it keeps pumping hydrogen molecules through, and then eventually electrons will be accepted by water, and then that's a product. It's a waste product of cellular respiration. So we don't need to know all of the different proteins and all of the different jobs that the proteins do, but I do want you to be aware that there are several different protein molecules, and they have different jobs, okay? So here's the good part. So now here's our intermembrane space where we've pumped all the hydrogens, and now they want to move back through because it's a really high concentration gradient. They want to get out of there. They want to go to the low concentration gradient, which is in the inside portion of the mitochondria, the matrix. So this protein, integral protein, is going to let the hydrogen molecules through, but it's not going to let them through for free. It's going to capture the energy, the electrical energy that they create from going from a very positive space to a neutral charged space. And as that happens, it's going to phosphorylate an ADP molecule to make ATP. So this creates a lot of ATP in the process, and again, ATP synthase is the molecule that is doing it. So the ATP yield is 30 to 36 per glucose molecule. So this last step, remember, glucose is no longer around. We've broken glucose up, so glucose is not even in this last step, but the products from breaking glucose up are, which are NADH and FADH2. We make about 30 to 36 ATP per glucose molecule. It ranges 30 to 36 depending on which organism you're talking about. Some organisms are more efficient than others. And to give you a very sad statistic or sad data, the cellular respiration, the overall cellular respiration, all the steps together, really only get about 34% of the energy stored in glucose. the rest of it is typically released as heat. So we're already releasing a lot of heat from glucose, even though we're doing it in small incremental steps. So imagine if we did it in one giant step, we would release so much heat and barely get any energy from glucose at all. Okay, so this is a very summarized version of the electron transport chain. Again, outer membrane of the mitochondria, inner membrane of the mitochondria. This is the folded one. On that membrane, there's a lot of these proteins. There's not just one of each, there's a lot. And this is where we're putting all those hydrogens. So we get that really high concentration of hydrogen ions, which are charged. They have a plus charge. And they can't move through the membrane on their own, because they are a charged molecule. They're an ion. So in order to get away from the high concentration, they have to go through this ATP synthase, and that allows the phosphorylation of ADP to ATP. Okay, so that's, again, the major goal of our process here. Okay, so if oxygen isn't present, why can't the mitochondria continue with aerobic respiration? Well, we need oxygen to accept some electrons at the end. Actually, oxygen will turn into water at the end. Where does the pathway stop if oxygen isn't present? Well, let's go backwards. So if we don't have electron donors here, and they're just locked up as NAD, FAD, they're not being utilized here, and pyruvate's not being made to acetyl-CoA, well, we can't do anything past the glucose point. So we can actually do a different step after glucose. Instead of going into a mitochondria, we can do something else, which is what we're going to get to next. Okay, so hopefully now you can talk about the electron transport chain pretty fluently. Try explaining it to somebody, explain it to your dog or whatever pet you have, and then explain how the hydrogen-proton gradient is important and why we need that gradient set up in order to create ATP. Like, what would happen if we didn't have that gradient? Okay, 7.5. So we're going to talk about what happens when we don't have oxygen. So the creation of acetyl-CoA, the Kreb cycle, and the electron transport chain occur with oxygen. So if we don't have oxygen, we're going to go a different route. So we'll talk about what fermentation is, and that's called anaerobic. Well, fermentation is a type of anaerobic respiration. So if you think back to glycolysis, glycolysis doesn't care if oxygen is present or isn't present. Oxygen does not have a role in glycolysis. So glycolysis, remember, it needs a few things to begin. It needs a glucose, it needs 2 ATP to start, and then it needs 2 NAD molecules. So NAD molecules come from the electron transport chain. When we extract the energy from NADH, we end up with NAD. So NAD is needed for glycolysis to happen, for glycolysis to move forward. So if we're not generating NAD from the electron transport chain, we need to generate NAD in a different manner. So we're going to do fermentation. So fermentation will take those electrons off of NADH and create NAD. So there's 2 types of fermentation. There's lactic acid fermentation and alcoholic fermentation. And I do want to point out again that anaerobic respiration, which is respiration without oxygen, so glycolysis and fermentation, is not that powerful. It doesn't create that much ATP, but at least it creates some, so it's better than nothing. So again, glycolysis happens in the cytosol, doesn't care if oxygen is present or isn't present. It's going to happen anyway, as long as we have our reactants. which are glucose, NAD, and ATP. So if no oxygen is present, we do fermentation so that we can regenerate NAD. So the first type of fermentation we'll talk about is lactic acid fermentation, which is what we do. Lactic acid fermentation occurs in the muscle cells, usually when oxygen is limited. So when you've gone for a long run or you've been playing sports for hours at a time, your cells will start to utilize lactic acid fermentation because they're just not getting enough oxygen. So it's not a bad thing. They need to do it in order to generate NAD again, and this is how they do it. So you have glucose in your body that goes through glycolysis and creates 2-pyruvate. Fine, we're used to that, right? So after this point, 2-pyruvate, instead of them being formed into acetyl-CoA, they go through fermentation and they're made into lactate instead. And that production of lactate uses up some of the electron energy created in the glycolysis step. So remember, glycolysis makes those NAD molecules, NADH molecules that are used in the electron transport chain. But since oxygen's not available and it's not driving the electron transport chain, it's halted it, we have to generate our NAD again some other way. So we take NADH and we break the bond off, and we use that energy to make lactate instead. Alcoholic fermentation is another way that we can create NAD from NADH. So instead of pyruvate becoming acetyl-CoA, it becomes acetylaldehyde, and that acetylaldehyde is made into ethanol. So alcoholic fermentation releases CO2. So when you bake bread in the oven, what happens? It creates those pockets of air, or you might just see them as holes in your bread. But anyway, they're pockets of air, and it's created by yeast undergoing alcoholic fermentation. So as it's releasing CO2, it creates like a pocket of air around it, and then it creates ethanol as well. But that ethanol is usually just evaporated from the heat. So alcoholic fermentation has 2 steps. The second step is where NADH is made back into NAD, and that NAD goes back into glycolysis. So anaerobic respiration is not exactly the same as fermentation. Anaerobic respiration is kind of an umbrella term, and that's what happens when we don't have oxygen. So some living organisms can use an inorganic molecule as a final electron acceptor in the electron transport chain. So this is pretty cool because instead of oxygen, they're able to use something like nitrous oxide, sulfate, and iron instead. So their Kreb cycle and their electron transport chain can continue on normally because they're using these instead of oxygen. So they're actually still creating a lot of ATP, and they're still making ATP through chemiosmosis. They're just doing it a little bit differently. So some organisms can do it, not many. A lot of the time methane is produced, and that creates that really smelly, swampy smell after the rain when you go through the swamp. So just think about that next time it rains and you go through the swamp and you're like, oh, it smells. Anaerobic respiration, that's what's happening. Okay, there's a nice video from Bozeman Science that goes over anaerobic respiration a little bit more if you want to watch that. But that's the end of this section. We have one more section to cover. Different pathways. So we've talked about glucose as the key molecule here for what we're extracting energy from, but we can use proteins and lipids as well. So not just carbohydrates, but we can use parts of proteins, amino acids, glycerol. Glycerol is part of what? fatty acids, right? It's that glycerol head and then the 3 fatty acid tails. So glycerol can enter glycolysis as well as some amino acids. Some amino acids can enter pyruvate, the pyruvate oxidation, acetyl-CoA step. Some fatty acids and amino acids can enter the citric acid cycle, so they can like skip the line. And then nothing else can really enter phosphorylation. We need to create those electron carriers in this step so that we can use them in phosphorylation. But carbohydrates, amino acids, remember, are part of proteins. Glycerol is part of lipids. Fatty acids are lipids. So these are just different ways that the body can utilize different biomolecules, macromolecules. And it's interesting because we can adapt to different situations. So if we're not eating a lot of carbohydrates, but we're still eating like a lot of meat and fat, and we're getting our protein and lipids from the meat, we can still create ATP. So that's kind of why the keto diet is so is able to still function, because you're not eating carbs, right? You'd think that you wouldn't be able to function, but your body can do different pathways. So these are just different ways in different areas where amino acids can enter the pyruvate and the acetyl-CoA step and the citric acid step, but we don't need to memorize them. It's just something interesting to note. Okay, and that's the end of 7.6. So just know that proteins, lipids, they can enter this pathway as well, but they don't always enter at the beginning. They can enter at different parts of the overall process. So hopefully now you know what cellular respiration is. You can tell me what glycolysis is. You can tell me what fermentation is and why fermentation might happen. And then you can tell me what happens when oxygen is present and we do acetyl-CoA, citric acid cycle, and electron transport chain. Okay, so that is it. I hope that was helpful. Have a great day.